Hello everyone, good evening. Good evening, Sudeshna. Good evening, Parth. Let's start with our today's class. Good evening, Akshita. Good evening, everyone. Good evening, Paras. Is the screen visible? Good evening, Vaibhav. Okay, so good evening, everyone. My name is Muskan Kashyap, and I am the founder of Biotechnology, where I focus on teaching bioinformatics and computational biology in a very simple and a very practical manner. So, I will be guiding you through this entire 7-day NGS bootcamp. So, before we talk about bioinformatics or NGS, let's start with one basic question. And that one basic question is, why are we studying biology? So we study biology to understand life, to study about diseases, for research purposes, for career. So all of the biology, everything, it comes down to one thing, and that is DNA. Whether it's your height, whether it's your eye color, whether it's a disease or how your body functions, everything is controlled by DNA. So, DNA is like an instruction manual for your body, and DNA is written in a four-letter language: ATGC: adenine, thymine, guanine, and cytosine. So DNA is made of only these four bases: ATGC. So if we talk about human DNA has around 3 billion bases, means 3 billion letters of ATGC. Now, can you analyze these 3 billion bases manually? Can you read or can you analyze these 3 billion letters manually? No, we cannot analyze these 3 billion bases manually. So this is the biggest challenge in modern biology, that we can generate the DNA data, but we cannot analyze it manually because it is too huge, it is too large. So with modern machines, we can read the DNA very fast. Here we have one problem, that we have the DNA data, but we couldn't analyze it manually because it was too large. So we solved this problem by producing the modern machines which can read the DNA very fast. So we solved our problem on how to analyze the data. But now we have another problem, that these machines, these modern machines, they generate huge amount of data. So we needed a way to handle this data. So bioinformatics is the field where we use computers to analyze this biological data. So bioinformatics is a combination of three things: biology, where we understand about DNA; computer science, where we write our programs; data analysis, where we handle large data. So bioinformatics is a combination of biology, computer science, and data analysis. So what is bioinformatics? Bioinformatics is the field where we use computers to analyze biological data. So today, bioinformatics is used in cancer research, it is used in drug discovery, it is used in personalized medicine, it is used in genome projects, it has been used in COVID variant tracking. So that's why bioinformatics is one of the fastest growing fields. And the most important part is that you do not need to be a hardcore programmer to start in the field of bioinformatics. You just need some basic biology understanding, you just need some step-by-step guidance and a little practice. So now we understand that DNA is important, the data is huge, we need computers and we use bioinformatics to analyze the data. But the question is, how do we get this DNA data to these modern machines? And where does this DNA data come from? Do we directly get this DNA data into these modern machines? No, we don't directly get this DNA data into modern machines. So, firstly, we need to extract the DNA. So what do we need to do? First of all, we need to extract the DNA from a sample. So we take the sample like blood sample, we can take saliva as a sample, we take tissue sample. So from this sample, we extract the DNA in the lab. So from these samples, we have extracted the DNA, we have the DNA now, but can we read it directly? We extracted the DNA here from these samples, but can we read this DNA directly? No, we cannot read this DNA directly. So to read this DNA that we extract in the lab, we use a process called DNA sequencing. So sequencing simply means finding out the exact order of ATGC. We understand that DNA is only made of these four bases A, T, G and C. These four bases make millions and billions of sequences. So we want to find out the exact order of this ATGC inside the DNA, and that process of reading the DNA is called DNA sequencing. But here's another problem, that the DNA is extremely long. As we have talked about the human DNA, that the human DNA is about 3 billion bases long. We are talking about in billions, 3 billion bases. So for example here, this is a 3 billion bases long DNA. So we cannot read it at once because it is too large. So we cannot read the DNA, we cannot find out the exact order of ATGC in a DNA which is 3 billion bases long at once. So what scientists have decided to do is very smart. So scientists, they take the DNA and they break the DNA into small pieces. So here in the lab, we extracted the DNA. So scientists break this DNA into small pieces. Now each small piece of DNA is called a read. What is it called? It is called a read. So what is a read? It is a small fragment of DNA sequence. So here what we have done till here, we took the sample, we extracted the DNA. Here we broke the DNA into small pieces and these small pieces of DNA are now called reads. So now we have reads. So what is our work now? That we have to read, we have to analyze these reads, these DNA fragments. So now we need a machine that reads these small DNA pieces, and that machine, that technology is called the NGS, which stands for Next Generation Sequencing. So NGS is the technology that reads millions of DNA fragments at the same time. So here what is happening? This is the NGS machine. For example, assume that this is the NGS machine. It reads the small DNA fragments. Okay, this NGS machine, it reads those small DNA fragments that scientists have cut into small pieces, and those DNA fragments are called reads. So here NGS, what output will NGS give us? Here, what input are we giving? We are giving the input of small DNA fragments, correct? And we are giving the input of millions of small DNA fragments, because here we are talking about 3 billion bases. Okay, DNA is a 3 billion bases long, which we have cut into small pieces. So here, this is the input that we have given to the NGS machine. So what output the NGS machine will give us? It will give us small reads, millions of small reads. Okay, so the NGS machine, it does not give us full DNA sequence, it gives us millions of small reads. So we are comfortable with this term reads, it only means small DNA fragments. So this is the output that the NGS machine gives us. So after sequencing, now what do we understand by the term sequencing? Reading the DNA that we given as an input to this NGS machine. So after this NGS machine read those small DNA fragments, it has given us these millions of reads as output. So means NGS machine has read the DNA and this is the exact order of the DNA that this machine has given to us. So I can say that after sequencing, what do we actually get? We get a large collection of reads. Now this large collection of reads is called raw sequencing data. What is it called? It is called raw sequencing data. So raw sequencing data simply means all of these reads because they are very large in numbers. So this raw data, all of these reads, they are stored in a file and that file is called the FASTQ file. So if we look at the workflow, what happened? First of all, we took the sample. From the sample, we extracted the DNA. From the extracted DNA, we cut it into small pieces, which is called DNA fragments. Then we gave it as an input to the NGS machine. So NGS machine read this DNA and it give us an output of the read. So what are these called? These are called reads. And the FASTQ file is where this output is stored. So DNA sequencing is the process of reading the DNA and NGS is the modern technology that performs that process of DNA sequencing at a very large scale. So here are some of the examples of the NGS machines. Now we have understood what is NGS. NGS is a technology, it is a method, it is an approach of DNA sequencing. And Illumina is the company that develops this technology and builds the machine. So these are some of the examples of the NGS machines which are developed by the company Illumina. So here you are seeing the first example, Illumina MI sequence, it is an NGS machine. Illumina next sequence, it is another NGS machine. Illumina Nova sequence, it is another NGS machine. So these NGS machines are the instruments that actually perform the sequencing. So are we clear till here about the FASTQ, that the output of an NGS machine is a FASTQ file, and the terms that we have read here till now? The read, what do we understand by the reads? The raw sequencing data, DNA fragments, FASTQ file. So are we clear till here? Do we have any confusion how the NGS machine gives us the output of a FASTQ file? Any confusion? Very good, Paras. Very good, Shraddha. Okay, Vanshika, Subhashri, Parth. Okay, very good. So now we will understand this FASTQ file. Till here, we understand. Yes, the output of an NGS machine is always in a FASTQ file. What we have understood by this workflow, that we extract the DNA, we cut the DNA into pieces, we give this DNA fragments to the NGS machine and NGS machine gives us the output of the reads, which we call the raw sequencing data. Now we need to save this data somewhere, right? So we save this data in a FASTQ file. Okay, so the output of an NGS machine is a FASTQ file. Is it clear? Okay, let's continue. So we are going to deeply understand about the FASTQ file, because in this course, in this 7-day course, we are going to learn how we can analyze the NGS data. And what is the NGS data? This is the row sequencing data which this machine has produced. Okay, these are the reads, and these reads are stored inside this FASTQ file. So what are we going to learn? We are going to learn how we can analyze the data, the NGS data which is stored inside this FASTQ file. Okay, so till here we understood that NGS, it reads, it analyzes the DNA fragments, it produces the data, and this data is stored in a file, and that file is called the FASTQ file. So let's see about the FASTQ file. So FASTQ file is actually a text file. What is a FASTQ file? FASTQ file is actually a text file. So now we understand that NGS machine give us the exact order of ATGC. And here what we did, these are millions of reads. These are millions of small DNA fragments which we have given to NGS to read. And NGS give us the exact sequence of ATGC also in the form of reads, which are millions in number. Correct? So here, if we are talking about a FASTQ file, so this FASTQ file, it contains millions of reads. And by millions of reads, we mean millions of DNA sequences. So in this diagram here, you are only seeing a single read. Okay, it is only a single read. Because all the reads are the same, they all contain the DNA sequence. So we are going to understand it with an example of a single read. Okay, but always remember the FASTQ file contains thousands and millions of reads. So here, what does this FASTQ file store? First of all, this FASTQ file will store the reads, and in the reads we have the DNA sequence. So this is the first thing that this FASTQ file store, the exact order of ATGC, the DNA sequence. And the second thing that this FASTQ file store is the quality of each base. Now, what do we understand by the quality of each base? So here we are saying that this is the sequence ATGCCCGTAGCTA. This is our read 1. This is our DNA sequence 1. So this is the exact order of the DNA sequence that NGS machine has provided us. So here, this quality score, it means that how confident the NGS machine is that this base is A. For example, the NGS machine is 100% sure that this first base is A. Now let's come on to the base T. Here the NGS machine says that the NGS machine is almost 60% sure that this base T is correct. Here, let's see the G base, which is the third base. So here the NGS machine is saying us that NGS machine is only 10% confident that this base is G. So what are we talking about? What are we saying? That NGS machine is telling us how confident the machine is that this base is actually 100% correct. This base is actually 50% correct. This base here is actually 10% correct. So what is it? It is the quality scores. Okay, we are going to learn about quality scores in depth, but for now we are only understanding that this FASTQ file stores two things. First of all, the DNA sequence, and second of all, the quality scores. And each base has its own quality score. Now, let's see about this read again. So here, this is another read. Again, it is one read. Okay, it is only a single read that we are looking at. So inside of a FASTQ file that we have already talked, the FASTQ file contains millions of reads, and here it is a diagram of a single read. Now, each read, okay, each read in a FASTQ has 4 lines. So first line, the FASTQ file starts with the @ or I can say that a read each and every read, they starts with the @ Okay, so if you see the @, it means it is starting of a read, it is starting of a FASTQ file. Okay, so after the @, the thing that you are seeing here written, it is called the sequence ID. What is it called? It is called the sequence ID. So this is the first line. The starting of the @ means it is a FASTQ file. Then after @ we have the sequence ID. So this is the first line. In the second line, we have the DNA sequence, the actual DNA sequence that we will analyze further in this course. Okay, so this is the actual DNA sequence in the second line. In the third line, we have this plus sign. So this plus sign is actually called the separator. What is it called? It is called the separator. So it has no biological meaning. It simply means to separate these two. So here, this is the actual DNA sequence, and here in the fourth line, this is the quality scores. Okay, so what does this separator do? It only separates this DNA sequence and this quality score. So each read, for example, there are 5,000 reads in a FASTQ file. So it means there are 5,000 DNA sequences, and each base will have its own quality score. And each read is of 4 lines. The @ the sequence ID, in the next line the DNA sequence, and the plus sign, which is the separator, and in the fourth line we have the quality score. So now here the quality score, it is actually the most important part to understand in the FASTQ file. Let's look at this example here. So as we talked about here in this example, that NGS machine tell us that NGS machine is 100% sure, very sure that this base A is correct and it is A. But next, for the next base, the NGS machine says that it is only 10% confident. Only 10%, from 100 to 10% that this base is T. So now, can you trust this base? Can we trust this base because its quality score is very low? Which means that NGS machine is trying to say that NGS machine is very underconfident, very low confident that this base is actually T. So now, can we trust this base? We cannot trust this base. For example, you want to find out the mutation. For example, you have a patient and you want to find out the mutation in your patient DNA, and then you want to suggest some personalized medicine. So here, if you give the data of the DNA to the NGS machine, and the NGS machine reads that DNA and it gives you this sequence, this exact ATGC sequence. But here the NGS machine is saying that it is not sure that whether this G, here T, here A, whether they are 100% correct. Okay, so NGS machine gives you confidence in the form of quality scores. So if you don't trust whether this G base is correct or not, it means your primary data, this will be your primary data to detect the mutations, which means your primary data is wrong, which means you will have wrong mutations. If you have the wrong mutations, you will suggest wrong personalized medicine, right? So here, what are we talking about? That this NGS machine is not 100% accurate. Okay, this NGS machine is not 100% accurate. These machines are not perfect. So what does this machine say? This machine, it reads the DNA, then it gives us the sequence, and here, this machines, sometimes it can read A as A, but sometimes it can read A as G. So here, what is happening? The machine tell us along with this DNA letter. First of all, this machine give us this DNA letters. So along with this DNA letters, machine is trying to say with the quality score that it is this much confident, 100% confident, 20% confident, or 50% confident that these bases are correct. Okay, so here we cannot fully trust the sequence given by this NGS machine. Okay, so if we cannot trust this sequence, we cannot go for downstream analysis. Okay, so along with this DNA sequence, machine, this NGS machine, also tell us how confident it is about each letter, and that is called the quality score. So here, let's look at the workflow again. What is the actual output of the NGS machine? We took the sample, we extracted the DNA from the sample, we break the DNA into small pieces, we give it as an input to the NGS machine. Now, what NGS machine is returning us? It is giving us an output, first of all, a DNA sequence and the quality scores of each base, which is stored inside the FASTQ file. So now we are understanding what does FASTQ file store. It stores millions of reads and millions of quality scores. Now look at this example here. Now we are going to understand the quality scores. What are these? How do we understand it? So here, look at this. For example, this is the DNA sequence that the NGS machine has given to us, and this is the quality score. So here, this is I. Okay, it is the I character. So here, the NGS machine is saying that this is the sequence, this is the DNA sequence, and this is the base A. So for the first base A, NGS machine has given us the quality score I. Next, for base T, the NGS machine has given us the quality score I. Means here, for all bases in this sequence, the NGS machine has given us the quality score I. Now, what does this I actually mean? So, whenever you see I character as a quality score, it means that the NGS machine is very sure, we can say almost 100% sure that this base A is correct. Now, each of these bases have the quality score I, which means NGS machine is very confident that all the bases are correct. But again, what does this I character mean? So, for example, understand this example. It is a very simple example. In the school system, in the school we have the grade system, right? If you have marks between 91 to 100, you will have the grade A. If you have the marks between 61 to 70, you will have the grade B. If you have the marks between, for example, 41 to 50, you will have the grade C. Or if you have marks 5, you will have the grade F. So what is it? It is the grade system. All the grades, they represent some numbers, correct? So in the same way, these are the characters. Now these characters, which are called quality scores, they all represent a number. What do they represent? They all represent a number. Now for example, here this I character, its quality score is 40. Okay, these are the quality scores, means these are the characters, and this I character, its quality score is 40. So as I said that if you see I character, it means that the NGS machine is almost 100% confident that this base is correct. So here, what is the score of this I character? It is 40. So instead of writing 40, 40, 40 like this, what are we doing? We are storing these quality scores in the form of characters. So the FASTQ file, okay, the FASTQ file, it stores the quality score in the form of ASCII characters. So what are these characters called? These characters are called ASCII characters, and these all characters represent a number, that is the quality score. Okay, so are we getting something? We are understanding something about the quality scores. Now look at this another example here. So now you can see there are some different quality scores. Here we have the sequence, DNA sequence, NGS machine give us this exact DNA sequence. For example, this is a read 1 in a FASTQ file, and this is the quality score of this read. So here we have already understood that if we see the I character, it means that the NGS machine is very confident, 100% confident that this base is correct. But if you see a exclamation mark, what are these? These are all the ASCII characters. If you see the exclamation mark, it means the NGS machine is saying that NGS machine is almost 5% confident that this base is C. Can I say here that it totally means that this base C is wrong here? right? Here we learned about two characters, one character I and another ASCII character exclamation mark. Okay, so if you see exclamation mark, it means that the NGS machine says that NGS machine is very underconfident that this base C is correct, which means that this base C, there are 95% chances that this base is wrong. So these are the quality scores that tells us about the sequence, that if they are correct or not. So just like we talked about that the NGS machine is not perfect, it can read A as G or C as T. So here we are seeing what quality score tell us. They tell us how NGS machine, how confident the machine is about these exact order of ATGC. Now, the important part is, as we are saying it again and again, that a FASTQ file contains millions of reads. For example, this is the first read, same second read with its quality score, third read with its quality score, fourth read with its quality score, and so on. So there are millions of reads, which means there will be millions of quality scores. So here you are only seeing some of the ASCII characters, but they are not enough. Okay, there are many ASCII characters. There are many ASCII characters, which means they have their own meaning, and we will have a million of DNA sequences, and each base will have its own quality score, means its own ASCII character. So this is not practical enough for us to learn about each character, to learn about each quality score. Okay, it is not practical enough, it is not manually possible for us to read each quality score for each base. Okay, that is where we are going to use the tool FastQC. Okay, we never memorize these characters, but you can study about it, you can build your knowledge, but we never memorize these characters because there will be millions of reads, each base will have its own character, means millions of quality scores. So we never memorize this, we never learn this ourselves, we use the tools. What do? We use the tool to understand about the sequence, whether the sequence, the quality of the sequence is good or bad. So we are going to use the FastQC tool, which is going to read our FastQ file. Means a FastQ file, for example, a FastQ file contains 5 million reads. So that FastQC tool will read our FastQC file and it will give us the report about all the reads in that FastQ file. Okay, so here, what do we understood here? We understood that our primary focus in the FastQ file was about the DNA sequence, that the FastQ file stored the DNA sequence, and along with the DNA sequence, it stores the quality scores. And one more important thing to remember here, sometimes we tend to forget it. For example, here we have 1, 2, 3, 4, 5, 6, 7, 8. These are 8 bases. Okay, 8 bases means we will have 8 quality scores. This is a very important rule, that sequence length, this is the sequence length 8, it must always match the length of the quality score. For example, you have the quality score only 3 and you have the bases 8. So your sequence length is 8, but here the quality score is only 1, 2, 3 IHF. Now, how are you going to tell what is the quality of these bases? We cannot tell this. So that is why the length, the sequence length must be equal to the quality length. That is the most important rule, because without one-to-one matching, here what are we doing? We are doing one-to-one matching. So without it, we cannot know which base is reliable, which is the quality of the base. Okay, so our primary focus here was to about understand the FastQ file. So we understood about the FastQ file and we understood about the quality scores. Do we have any question regarding the quality scores? Do we have any question regarding the quality scores? Krishna, that is what we talked about here. Look at here. That is what we talked about, that we have many ASCII characters. So we only talked about the highest and the lowest. Okay, I means 100% confident that NGS machine is saying that this bay is correct. Exclamation mark is that this NGS machine is very underconfident, very low confident that this base C is correct. Okay, so there is 95, or you can say 99% chances that this base C is wrong. So there are many ASCII characters and we do not read it manually, we do not read it ourselves, we do not analyze it manually. So that is where we are going to use the FastQC tool. Manasvi, the most common NGS machine used. Here, look at the example here. I showed you the example. These are the most commonly used NGS machines, okay, of the Illumina company. Okay, this is the MI sequence, this is the next sequence, this is the Nova sequence, all are from the Illumina. Okay, these are the most commonly used NGS machine. Shabnam, what's the difference between FastQ and FastQC? Shabnam, look here. In a FastQ file, we only have a DNA sequence. Okay, FastQ file is also a text file, and here FastQC file, it is also a text file. Okay, so in a FastQ file, we have only the DNA sequence. But in the FastQC file, along with the DNA sequence, we have the quality scores as well. So this is the output of the NGS machine. Darpan, yes, they are stored in the FastQC file. Krishna, here we were only taking the example. Okay, this does not necessarily mean that we will only have a sequence which will have the length 8. We can have the sequence which will have the length of 500 bases. We will have the read whose length will be, for example, 990 bases. So if we have a sequence whose length is 500 or 900 bases, that means we will have the quality score about the same length. Okay, there are fixed number of ASCII characters, but we do not need to memorize it because we are going to use the tool which is going to help us understand this FastQ file. Okay, I think I'm getting confused about the FASTA file. Someone was asking about FASTA. Yes, Shabnam, FASTA file, in the FASTA file is different, FastQ file is different. Okay, if I say FastQ file, if I say FastQC file, it is the same meaning. Okay, FASTA file is different and here FastQ file is different. It is the same thing. If I say FastQ file, if I say FastQC file, it is one thing. Okay. Manasvi, in the FASTA file, we only have the DNA sequence. Okay, here this is the DNA sequence. So a FASTA file will only have this DNA sequence. But a FastQ file, or should I say a FastQC file, okay, it contains the DNA sequence along with the quality scores that we just discussed. And here this FastQ file, or again, if I say FastQC, it is the same meaning. It is the output of this NGS machine. Okay, so in FastQ file, we have the DNA sequence along with the quality scores, that how confident that the NGS machine is that these are the exact ATGC bases. N means that the NGS machine does not know which base that is. It can be some contamination. Okay, we already know that we only have 4 bases, ATGC. So if there is some contamination, the NGS machine can give us N. For example, here we can have the sequence ATNCCNGTN. Okay, okay. So now we understand what the data looks like. And what data are we talking about? We are talking about the DNA sequences along with the quality scores stored inside the FASTQ file. Okay. So now we understand what the data looks like. But the next question is, how do we actually work with this data? We have the FASTQ file. Just imagine that you have a FASTQ file. Now, how will you work with that FASTQ file? We cannot open or analyze the FASTQ file using the normal windows like Word or Excel. Okay, we cannot use Word or Excel. So that is where we use Linux. So in bioinformatics, we use a system called Linux to work with large data like FASTQ, because now we understand a FASTQ file contains millions of reads, means a FASTQ file is a very huge file. Okay, so now we understand the FASTQ, it is a huge file with millions of reads. So to handle such large files, we need a powerful system, and that powerful system is Linux. Okay, so that is why we use Linux in bioinformatics. And here, Linux has many distributions, it has many versions. So we are going to use Ubuntu. Okay, so Ubuntu is a beginner friendly Linux. So that is why we are going to use Ubuntu. So I have already shared the link how to install Ubuntu in your system. Kindly visit that link of the YouTube channel and install the Ubuntu via WSL. It is all explained in that video. Okay, so let's open our Ubuntu. So this is the Ubuntu. Let's open it. So here we are seeing something written like this. So whenever you open the Ubuntu for the first time, so this is always shown. Okay. Yes, Madhulika, you can work on it. So whenever first time you open your Ubuntu, it will always show it like this. We can just close it and open it again, then it will just get removed. Let's open the Ubuntu again. Let's open it. Okay, so here we can see we are only seeing this. Now, this black screen that you are seeing, it is called the terminal. What is it called? It is called the terminal. So this is where all bioinformatics analysis happen. Okay, so now instead of just looking at the FASTQ file, we will actually open it and we will actually work with it using Linux. Okay, but today we are going to understand the most basic commands in Linux to work with the FASTQ file. So in the next session, we are going to work with the FASTQ file in Linux. So today we are going to learn some basic commands. So the first command that I am going to write is pwd. So this is the first command that I have written. Now what does this pwd means? It means print working directory. What does it mean? Print working directory. So this command will tell you that where are you in the system. Okay, it will give you the exact location that where are you in your computer. So let's press enter. So here we are seeing that I am inside home and I am inside this directory. Now where is it? Where is this home and this directory? So let's open my file explorer. Let's scroll down. So here we have Ubuntu. This is the home directory. Here we are seeing it that where are we? We are inside the home directory, and inside the, if I say directory or if I say folder, that's the same meaning. Okay. So don't get confused between directory and folders. Okay. If I say I'm inside the home directory and I'm inside this folder, so it means the same thing. So here is the home directory or the home folder. Let's open it. This is the folder. This is my folder. So let's open it. So we are inside this folder. So here, this is what this command is showing us, that where are we? We are inside this directory. So what I'm going to do, here we can see that inside this folder, I have some another folders, right? So if I want to list all these folders, for wait a minute. So here, these are the normal folders, okay? These are actually the hidden folders. We are not going to talk about this. We are going to talk about this. These are the normal folders. Just the normal folders that we create in our desktop. These are the normal folders, okay? So if we want to list these folders inside our terminal, we will write the command ls. So ls means to list all the folders, all the files inside the directory we are in. So let's press enter. So here we can see this is the folder FastQC, this is the folder, this is another folder, this is BWA folder, and this is another folder. So can we see these folders here? We can see it. Okay, so the ls command, it will show you all the folders and all the files that we have inside our working directory. So this is my working directory. Now, here we have seen that we have all these folders. Now let's create a folder of our own. Okay, I want to create my own folder where we will work in the from tomorrow when we will download the FASTQ file, when we will work with it. So we need a folder. So to make a folder, we have the command mkdir. Okay, mkdir, it means make directory. MK is for short for make, dir is short for directory. So mkdir means make directory. So here we have written the command to create the folder. Now I will give the name of my folder. So let's give the folder name NGS. So let's press enter. Now what we will do, again we will type the command ls because we want to see all the folders inside our working directory. So let's type the command ls, press enter. Now can we see this NGS folder that we just created here? So let's see it, let's refresh it. So here this is the NGS folder that we just created inside our terminal. Now what are we going to do? We are going to go inside this folder. Okay, we are going to go inside this folder. So here we have the command cd. CD means change directory. What does it mean? Change directory. So for now I am inside this directory, means I am inside this folder. Now I want to change my directory. I want to go inside this folder. So we will write the command cd and then we will type the name of our directory that we want to go inside. Now press enter. Now here you can see that now you are inside this NGS directory. So let's write this pwd command again. Print working directory, press enter. So now you can see that here when we type the command pwd, we were inside this directory. Now here when we type the command pwd, we are inside this directory and we can also see it here as well. Okay, so we created our own folder of NGS and now we are inside this. Now inside this folder, if we want to create more folders, what we will do, again we will just write the command mkdir. Now for example, I want to create separate folders for the data and I want to create separate folders for the result that will get generated. So here let's write data and let's write results. So here we have given the command mkdir, we have written the name of our folders. So here I have given space here. So if you give space here, it means this is another folder and this is another folder. If you do not give space, it means it will only be a single folder with the name data results. Okay, now let's press enter. So now what are we going to do? We are going to see all the folders inside this directory. Let's write the command ls, press enter. So now we can see inside this folder we have two folders. We have data and we have results. So let's check it. Let's go inside this and here we have data and here we have results. So let's go inside this data folder and let's create a file. So how do we go inside this data folder? This is the data folder. How are we going to go inside this? we have to change our directory. So let's write cd, let's write the folder name, press enter. Now you can see we are inside this data. Again, we can use the command pwd to check. So here we can see we are inside this data directory. So now I am inside this folder. Let's open it. So now we can see for now this folder is empty. So we are going to create a file. So to create a file, we have the command touch. Okay, touch is the command that you use to create a file. Now let's write the file name. So for example, my file name is test.txt. So to create a file, we will use this command. Now press enter. Now let's again write the command ls, press enter. So we are inside this data folder. Now inside this data folder, we have this text file. Let's check it, refresh it. So here we can see that we have created a file inside our terminal. Okay. Now, what are we going to do? Let's write something inside this file. Okay, because we are going to see another important command, and for that command we need to write something inside this file. So to write something inside this file, we will use the command echo. Okay, what command do we use to write inside a file? Echo. Now write in quotation marks whatever text you want to write. For example, I want to write hello. Let's write my name, Muskan. So the text that you will write inside this file, you will write it in quotation marks. Okay. Now here you have written the command that you want to write this text, but you have to also mention that where do you want to write it. So we want to write it inside this file, because here this folder data, it can contain many files. For example, just imagine that this data folder contains 5 files. Okay, and here we can see we are inside the data folder. So if we just simply write echo hello Muskan, echo our text, so you will be able to save this text in your required file. Okay, so that is why here we have to mention that in which file we want to write this text. So for that we will add this symbol and we will write the name of our file, test.txt. Okay, this is our file name, test.txt. Now press enter. Now what are we going to do? We are going to see our file. We have already written one line, one text here inside our file. So we are going to see our file. Okay, so here we have the command cat. If we want to open our file and if we want to see all the data inside the file, so we have the command cat. Okay, so let's write the name of a file, test.txt, and press enter. So here what happened, all the data that was saved inside your file is printed here. Okay, so what does this cat command do? It prints all the data that it is in your text file. Now if I want to write something like a paragraph, okay, I don't want to use the command echo again one at a time. I want to write something like a text editor, just like we have a notepad and we write it simply. In the same way I want to write it. So here we have one of the most important command, that is nano. Nano is a very important command. I am only showing you with a very simple example that I want to write something. But we are going to use this command if we want to see our FASTQ file, if we want to do some changes, we want to open it. So nano is a very important command. Okay, so we are only understanding it with a simple example. So here we have written the command nano. Let's write our file name, text.txt. Now what this command is going to do, it is going to open a simple editor where you can type like a notepad. So here as soon as I press enter, let's press enter. So now here you can see your test.txt file and this is the content inside your file. So let's write something here. For example, I want to write I am working in Linux. So we can write it like this, like a notepad. Okay, here let's write something that I am using Linux via WSL, or we can write in Ubuntu. Okay, whatever that we want to write, we are writing it like a notepad. So now what we have to do, we have to save this. Okay, so to save this, all of the commands are written here. So to save it, here it is written write out. It means Control O. If you want to save the data, we will press Control O. So let's press Control O. So now you can see file name to write text.txt. Yes, we want to write it inside this file. So press enter. So here now this data has stored inside our file, and here we can see wrote 4 lines. Okay, now to get out of this notepad, here we can see it is written Control X to exit, Control X. So everything is written here. So let's press Control X. So now we are outside of that text editor. So these are one of the most basic commands: pwd, ls, mkdir, or cd, or here cat or echo, and nano. It is one of the most important commands in Linux that we are going to use, that we use to navigate and work with the files. So from tomorrow, we will use these commands to actually analyze the data. And along with the analyzing the data, we will also learn new commands as well. Okay, so for now, practice these commands, install your Ubuntu, open your terminal, practice these commands, make your own directory. Okay, and from tomorrow, we are going to use these commands to actually analyze a real FastQ file. Okay, so any questions? Okay, so that was for today's session. Okay, very good. So you can leave the class if you do not have any questions. Thank you, Manaswi.